In this video, we will discuss about validation of 3D cell culture model. 3D cell culture model that they have developed, where they have used this GelMa of different concentrations, around 5% GelMa and 7.5% GelMa concentrations, and compare it with the collagen cells cultured on collagen 1. And for this comparisons, they compared the qPCR results of ECM related proteins, secretome related, not proteins, ECM related genes, secretome related genes using qPCR, and then compare these results with the transcriptomic results that they have got previously, that we have discussed in the previous, when we are discussing the previous results. So in the case of, let's start this ECM, it is extracellular matrix related genes. So here we can see that for BxPC3 and PANC-1, the CCL, the COL1A1 is expression levels are similar for BxPC3, but for this PANC-1 they seems to be slightly higher. Similarly, for this Col11A1, they see the similar trends that for collagens. In the PANC-1 case, it seems to be higher, but for CTSB, it remains similar. For the case of BxPC3, the FN1 expression levels are higher in the GelMa condition, 3D cell culture conditions, whereas it is lower in the case of PANC-1 condition in the 3D cell culture. When the PANC-1 cultured in 3D, and the PANC-1 cultured in the GelMa based hydrogel compared to the collagen. And for this MMP9, they see almost like similar results, as the difference is not presently significant. Similarly, for TGF-beta1, results are similar, and the VCAN and YAP1, here also the results look similar. That is, cell expressed in the respective GelMa based hydrogel is similar to the gene expressed in the GelMa based hydrogel is similar to the gene expressed in collagen in their respective conditions. Thereafter, they check the secretome related genes. Here also they see in most of the cases the secretome related genes are similar. Secretome related genes are similar to secretome related genes are similar for BxPC3 case, PANC-1 case, and it is in line with the cells that are cultured. These respective cells are cultured in the presence of collagen 1. However, the changes that they have seen is that the IL-6 is expressed high for the PANC-1 collagen. IL-6 seems to be expressed high for these collagen conditions compared to the other conditions. And thereafter, they also see the similar trend for IL-8. So IL-8 results are they have presented separately in this panel C, where we can see that for the collagen and for the BxPC3 and also for the PANC-1. For the collagen conditions, the expression levels of IL-8 is higher, but the expression levels in the 3D cultured GelMa based hydrogel is low for both BxPC3 and PANC-1 and for both the conditions. And this IL-8 is also known as the cytokines that helps in the pancreatic cancer progression. So, and in the case of the secretome related genes results in the heat map, the IL-6 levels, we can see that they seems to be expressed in higher amount in the case of this collagen based conditions compared to the their respective hydrogel based conditions. That can be seen from here. The PANC-1 conditions also shows the same thing. And so overall, they come to a point that there is a change in the gene expression levels depending on the stiffness around the cells. This is why they see different expression genes levels in the GelMa based hydrogel compared to the collagen, because the stiffness that has been observed by collagen based hydrogel is lower compared to GelMa based hydrogel that we have seen in the previous video. And for the IL-6, previously I have said that it expressed high amount, but the heat map looks like to show that for the collagen they are expressed in the lower amount compared to the other conditions. And but for this BxPC3, we can see that the expression levels looks similar to each other. So for PANC-1 the expression level seems lower. That's all for this video. Thank you for your time.